Hi and good afternoon, everyone. I'm Cyrus from Netrin Scientific. First of all, I would like to thank you for joining us today. Glad to meet everyone here. Today, we are proud to have this live webinar session by our honorable speaker, Dr. Joachim from Omni Life Sciences OLS, with title CASY, More in Cell Counting. Please allow me to make a brief introduction about Dr. Joachim. Dr. Joachim has completed his PhD study in Heidelberg in biochemistry field and started his career in BioRed. After 2 years working in sales team, he has been promoted to sales manager to take care for Germany, Austria, and Switzerland. He has continued his career as division manager for another 2 years before leaving for Honeywell. And in Honeywell, Dr. Joachim was responsible for lab chemical in EMEA area, and 3 years later has turned to global business manager for organic chemical. After some years, Dr. Joachim has transited back to life science in OLS to become manufacturer and focus on Asia region at the end of 2018 until present. Dr. Joachim has extensive experience about CASY from technical details to applications. Please don't be shy to ask any question if you have any, and you may also drop your questions on the chat box or Q&A session later. We will have the answer for you. So now, please welcome Dr. Joachim. Dr. Joachim, the floor is yours.
Thank you very much for a kind introduction. Yeah, I'm happy to do this call, and as you said, please step in, ask questions, especially during web meetings. It always shows that other audience is alive, so it's a rather good thing. I will touch. This presentation may be a bit different than normal presentation, as it's focused very clearly on benefits. As CASY is clearly a counter which has a different principle. So if some of the answers are not answered directly, we will touch a lot of applications, and I can also tell you a lot more about specific things, but I hope I can cover everything, because CASY is an instrument which can be used in a rather broad way, so for many things. You see my screen, I guess, so let's get started. If it works. Doesn't want. Oh, no. So quickly about OLS. We're a German company based in Bremen, which is north of Germany, and there we are having our office and producing the CASY and other instruments. That has started in 2015. Also, I heard some click. Not sure if there are questions already. Hello? That's okay, right? So because it's just a click from the app. So it's family owned, and where we do the manufacturing in Germany. And then we are exporting globally. Our major focus is around cell culture, cell counting.
I'm sorry to interrupt. I'm just sorry because I can't seem to see your slide.
Oh.
Okay, I'm not sure about others, but whether that is only me or others as well.
Let me see. I'll try again, but thanks for letting me know.
I'm so sorry. Okay, thank you.
Don't be. You should not
to interrupt you. Right. Thank you.
Better now?
I don't seem to be. Yeah. Okay. Yeah.
I can see your slides.
Yeah. Thank you.
Thanks for letting me know.
Thank you.
Good. So let's continue here. Anyway, so we're focusing on cell culture, on counting, on flow cytometry. We have on a distribution base, so we're also a distributor, and many other solutions. But let's talk today about cell counting specifically. And let me switch off my camera because I think my signal is better. So talking about counting, the question is what to expect, or what should cell counter do? And I think one of the most important things, obviously, is that it should count accurate and it should also count reproducible. So it should not be a number that you believe or not, it should be the number. And to achieve this, you always need some kind of statistical relevance. That means if you have 10 to the power of 9 samples, cells, whatever, the question is 1 times 10 to the power of 2 a statistically relevant sample? And many statistics people may say no. Another topic which many cell types are having problem with or presenting is aggregation, and that is through bacteria, through yeast, through mammalian cells, a common problem of which the counter should be or capable to handle. And then we have the cell by cell volume, which is also giving you information about the status of the cells or the level of changes, maybe contamination, so other additional information that may be of big value to many of our customers. And then finally, in some instances, like if you're related to pharma or other applications, you also may require GMP, GLP, or CFR 2157. So basically, why am I saying this? CASY can do that all. Now, the principle is very different, so you don't get a picture. That's probably the major optical difference that you will see, is that you will not see a picture. However, what you will see is a very precise size distribution. And this is a result of a very extremely simple principle of measurement. So what happened? Oh, why doesn't it work? Oh, now it works. So what happens is we have 2 electrodes, and these electrodes are separated by a defined pore. Now, when the system is idle, then the current flow here is constant, and we have a clear signal. Now, when the cell we measure, we pull cells through this pore, and we get a resistance. That resistance depends on the size, so the volume of the cell. And it's independent of orientation, as you can see here. Like we have this as an example, an elongated cell. If it passes a pore like this, then what we would get is a long signal, but a very flat one. As we measure this with a high frequency. If we just turn around and it would pass the pore like this, what we will get is a very high peak, but a very sharp one. What is now very important is, in both situations, the area of the peak is the same. So this principle, just to summarize, it makes it completely independent of shape, of orientation, of forms, whatever you have. Because, I mean, let's be honest, biology is not perfectly round-shaped. And that is true for mammalian cells, but if you look for bacteria, it's even more true. Like we have some customers, I'm not sure the microbiology people on this call may know them, like, what is this again? I forgot the name. It's a spiral bacteria, Leptospira, sorry, that was the name I was looking for. So used in animal vaccination, there are also some publications we have where CASY was used, which is a spiral form, which makes it very ugly to count. So CASY just does it because we are just looking for cell volume. Another example would be, we have some customers in Zurich, most known one are from Zurich, measuring amoebae, which would be another application where the form change, or trypanosomes or blood parasites. So it works with different shapes, with different forms quite easily. Is it just a number? No, it's not. So besides the pure number, you have a lot of additional information that helps you also to understand that this result is valid. So you always have full size distribution. So how is the breakdown of the different sizes of the different cells that were measured? In this case, it's human embryonic stem cells treated with acutase. And you see, yeah, this is a form, so it goes nicely up, goes straight down, so there's still some aggregation left. I can explain later on, next slide I think it is, how that works. We know how much cells were counted, like in this case, almost 6,000 from here to here. We did 3 cycles, so the instrument does 400 microliter per cycle. If we do 3 cycles, obviously we can also understand what is the standard deviation between the 3 cycles, which is another criteria for quality. Then we know how much viable cells we counted. We know what dead cells we had. We know how much debris we had. We know how much viable cells, viability, and so on and so on. So cell volume, biomass, it's all there, and much more that we present with every single measurement. Now the question is, how long does it take? Usually between 30 and 45 seconds, if not change from standard. Sensitivity, this is about, now for you maybe not transparent, this is, we can use a system with different capillaries, which are optimized for different cell types. So the 150 is the biggest one, which we usually use for blood cells, for like PBMCs, for mammalian cells, for big plankton, and we can go down to, in an optimized way, to 1,000 cells per milliliter. Standard would be 20,000. Same is true for 60, where we are looking majorly at yeast or algae or blood parasites, something like this. But optimized, we can also go down almost a factor of 10. And then finally, 45 is for bacteria and fungi spores and something very small. And there we can go down to concentrations like 2 times 10 to the power of 5. So actually, given that, it's very sensitive. And just keep in mind, especially for those working with bacteria, we are able to go extremely down with speed. So normally when you measure, you do plating, either you go plating, which takes a couple of days, or you do optical density, which compromises on the precision. You have precision in 45 seconds. So quite an adventure. What I already talked about at the beginning is, when you count, you want to rely on the result. And given that accuracy is gone, is given, the other thing is, how reproducible will it be? And this is an example which was done in Switzerland, and a customer called DSM, they do food nutrition stuff, and they were having a new instrument, and overall they were just doing a repeated measurement 6 times. Then they were looking at the result and like standard deviation of viability, of viable cell count, viability and peak. And I mean, I don't need to read the results yourself. This is something which is not faked, where we did not optimize the concentration of the cells or the preparation of the cells. It's just a real life result that you simply get whenever you use CASY. So to break it down to a very simple sentence, if you measure one sample 6 times, you have one result, not a variation of 20, 30, 40%. Good. Any question up to here?
Is there any question, anyone?
I do have one. Hello, can you hear me?
Yes, please.
Yeah. So far you talk about we are able to differentiate between dead cells and viable cells. So what if is it possible to look at whole blood cells, like when you have a mixture of different cells, and can you actually set a setting to differentiate different kind of cells, like just calculate like a particular type of cells in a cell mixture, or it doesn't do that?
The criteria here is clearly the size or volume of the cells. So I may have 1 or 2 examples later on.
Okay.
Which means it's like when we are looking at the PBMCs. I have an example. Give me 2, 3, 4, 5 slides. I don't know exactly the number.
Okay.
But we can also do like platelets or full blood.
All right. Okay.
I may have some examples. I think full blood is at the end, and PBMCs is somewhere in between. which is also the strength there because we can do multiple cursor areas. But stay with me a couple of minutes and probably this, I will answer your question anyway.
Yeah, sure. Okay. Thank you.
There was another question about bacteria, small bacteria I just saw on the line. Yeah, so as you can see, we accumulate the result, and if you look for, I think the question was if we can detect the bacteria, which is 0.1 micrometer times 6 micrometer. Now, what we are interested in is the volume, the total volume, and that was be expressed later on in basically in the size of a perfectly round-shaped object, because yeah, I don't want to go into too much detail. It has some just display benefits for displaying it. Yeah, we can do most of them. We can do. Yeah, so this, I think you mean rod-shaped bacteria, which have a small diameter but are quite long. Normally work very well. This is something, a slide which I try to create based on what probably most of you normally do, to see the difference, so to try to illustrate the difference. So you see a picture on the right side which has a very low resolution, and this resolution matches somehow how much counts you do normally optically or with normal counters, which is a couple of hundred. And the purpose is to illustrate you cannot expect that if you just count a couple of hundred cells out of 10 to the power of 6, 7, 8, 9, or even more, to see all details, to have a statistically relevant sample, or even to see contaminations or any of the other information that CASY will provide. For that, you need really high resolution. And this is given when you use CASY. So we are doing like 5,000, 10,000 mammalian cells, up to 200,000 bacteria in one measurement in a very short time. And that allows us to see more details, more like contaminations and many other things. And this also makes it extremely reproducible. I mean, just mathematically, if you have such a low reference number, besides other disadvantages, you will have high fluctuations in the result. It has to be. And you will for sure not see any contamination or anything. Now, the other thing is, just to illustrate it, what we do is we measure in 3D. So a cell is free floating around, is poured through a pore, and we have basically a 360-degree view, however you want to call it. So we are looking for the volume, and volume is a quite good representative information about size of a cell or of an object. And to be more clear, we guarantee that we do this with a precision of plus or minus 2% deviation maximum. And as said earlier, it's independent of shape and orientation, all these kind of things. So at the bottom line, what you have is extremely precise and reproducible information about the sizes of your cells or aggregates. And that allows us sometimes, many customers, and I have examples later, to see changes in the status of the cells. It can be starvation, where the cells grow. It can be baculovirus infection, where people stop the infection before the cells burst. So a lot of additional things that create, besides this pure count number, additional value. Or even like we have some testimonial from customers in bioproduction, they simply depend on CASY for many years now to standardize the process. It's an important one. If you compare that with anything optical, you may also get a size distribution, but this is not a size distribution, it's a diameter distribution. And diameter itself has some issues, like where do you focus? The focus level has a big impact on the diameter. And I can tell you, we know that this autofocus or manual focus is causing a lot of issues, not only on size, but also on viability. So where do you focus? Then if the cells sit longer on the slide, they may be a bit more bigger than if they just were entered. And then the final thing is biology is not perfectly round shaped. So what is the diameter if it's in case, for example, for an elongated cell? So it's another problem. So really it cannot offer any kind of good information as you have too much variables in there. Good. Now, why am I talking about sizes? Sizes can tell you quite a lot. So the first thing is it shows you how homogeneous your cells are. And this is, for example, important if you want to see, like, first one is T cells. And I have later on an example when you activate T cells, this changes dramatically. So T cells are normally quite sharp peaks, so extremely homogeneous. Then you have medium-sized peaks, which are, for example, fibrobust or Algae is used for beer production, or and so on. And then you may have broad peaks, which represent rather heterogeneous cells. And here is a good example, like MSCs. MSCs are extremely heterogeneous in size, and you can immediately see that when you measure the cells. Giving the top panel here, they are all homogeneous. So you see the peak goes up, the peak goes down. It's symmetric. Now, once this peak gets asymmetric, like down here, that's always an indication that you have aggregation. And I have a special slide on aggregation, and so I will cover that not here. But it's important not to ignore aggregates, because they are a significant portion of the cell population, and they are there. They're breathing, they're metabolism, they're doing metabolism, so they will do something to your experiments. So ignoring can be a very dangerous thing, as also aggregation is not a constant. We have a nice examples for this one. So many people look for changes in size. For example, one is the baculovirus system, but we have also people that look for impact of chemicals to the cells and so on. Another nice example is whenever you see shoulders or dips, it may be that you have a problem. Or no, that's maybe too much, but at least it shows you that you have another distinct cell population. In case of stem cell, that may mean you have unwanted differentiation, for example, or it may be wanted, don't know. However, it can be even in worst case, and I had that in a couple of times, and even one time in a shared demonstration with an optical counter, so side by side, where they said, yeah, we had good results. I had a problem because I saw something like this. So not only the peak that I was expecting, but a peak where I was not expecting it. And by default, whenever I have this situation now, and I also tell it to all my distributors and partners, measure the media. Now you can spend your own thoughts, what kind of cells you want to have in pure media. I would say probably none. So yeah, this group had a problem with contamination. I don't know exactly what it was, but usually in this case, it was around 4 or 5 micrometers, which normally is the size of yeast. CASY can tell you more. It can tell you how good your cells are, if they're aggregating, if they change for some reason which are wanted or not wanted. It can show you contamination, maybe by differentiation of things, but it can also show you differentiation, like if there's something wrong with the media or contamination, things are going on. Any question to this slide? Because this is a rather important one. Okay. Then I move on. It's known that optical, especially trypan blue counting, has sometimes issues with viability. And I'll come to this point, PBMCs soon. The point here is CASY has a very simple criteria for whether a cell is viable or dead. It's the cell membrane. And the cell membrane is intact, then the current cannot pass the cell. That means the full cell volume is detected. That's viable, and we get a peak for that. If the cell is dead, that means the cell membrane is not a barrier to current anymore. It's breaking. It's broken. It's ruptured. Sometimes it's even already gone completely, and we only have nucleus. I ignored ER and things like this yet here. And then it's dead. So it's a clear answer, yes or no. It's not like different kind of shades or something, which is important to understand. And there were publications about comparing this detection and how good it is. And just highlighting one here. And to make it a bit easier to read, I increase the size, as I will do for other slides as well. And let me read it to you. This is done from a group in Sweden. So not related to us, I did it independently, where they were comparing the results from counting, from analysis of MSCs, which have been cryopreserved before and after, before freezing, after thawing, and they were comparing both. And the result, I think it's on page 11 on top, if you would like to see, is we detect a higher degree of dead cells with both CASY counter and flow cytometry, as opposed with trypan blue staining, which potentially overestimates viable cell number. Obviously, I like this sentence, but it's also something we hear much more often, that with optical counting, you have viabilities of 80–90%, yet that's not always reflecting the real situation. And for CASY to be put in the same specificity or same precision as a flow cytometer, I think it could be worse. Yeah? So we like this to be in that same pocket with flow cytometry. Same time, we are much faster and much cheaper and much easier to operate. Good. Aggregation. I was thinking a long time how to make it visible, what that is, but the point is very simple. When you look at the market, most people are, and most competitors are ignoring aggregation. And the simple reason is they cannot fix it. So if you cannot fix it, best ignore it. However, we can fix it, and that's the reason why we should not ignore it, because it's a problem. The first attempt is what many people do, and if you look at the manual for a hemocytometer or something, they say ignore aggregates. Ignoring viable cells in your sample that add to the total cell count, to everything that happens downstream, for me seems to be a very dangerous thing, especially if it's not a constant. So the level of aggregation may vary even for the same cell, depending on the status of the cell and how they are. So it's not a constant that, I mean, if it would be then, you say, okay, I have a result and somehow I can extrapolate, but this is not given. So, and if you especially look for cells like iPSCs, it does not work. iPSCs are so heavily aggregating, and even if you singularize them, most of the cells will die and you cannot analyze them. And I have later on an example on iPSCs. I saw from China, for example, some people that were swearing that manual counting is the best for iPSCs. And then when I analyzed the results on CASY, I could show them that they killed 90% of their cells just to get some single cells to count. Yeah. So anyway, aggregation correction is not, well, ignoring aggregates is not an option. Now, the next alternative, which optically you can try to do, is draw some cycles. You can choose yourself if you're happy with that or not. And I try to visualize this with this artificial cells, but to be honest, I think we all would agree that it may work for small aggregates, like 2-threaded, 3-threads. But as soon as you have bigger aggregates, I mean, just think about that, 3-dimensional, you will miss most of the information. So it cannot work. So it's still something you try to overcome aggregation, but it's a ambient number. It doesn't work. So now what CASY does is a very simple, different principle. I told you earlier that we measure extremely precisely the volume of each cell. The same is true when we measure aggregates. We measure the total cell volume of aggregates. Yeah? And also with a precision of plus or minus 2%. And we do that with each measurement anyway. So everything that passes a pore is detected by volume. And then it's simple for us to analyze how often a single cell would fit into an aggregate. So it's fast, it's reliable, and it's also very transparent. Now, to make it a bit more clear what happens, we have this slide. On the left side, you see 4 different kind of aggregates, just for illustration, and some numbers. Now, just take a look at the first one, which is a single cell. Let's assume it has a volume of 524 femtoliters. Why that number? Because if you have that volume in a perfectly round-shaped object, it would have exactly 10 micrometer in diameter. Yeah. Now, you measure an aggregate, which is very big, and all of a sudden you have an object of almost 7,400 femtoliters. Now, it should be obvious that this cannot be a large cell. So the question is, how many cells are inside? And by this method, you would just count how often would this one fit in here. So this is 14 times. And once you activate the aggregation mode, the CASY would count this as 14 cells instead of 1. And we have this here from there, analyzing the effect of nutrition on cells on different. And there they are depending very much on the system because they need to give the information as concentration per cell. So they're using this aggregation correction quite heavily. Good. Let's give another practical example for aggregation. And this is obviously iPSCs. They're aggregated, you know them, on a plate. So usually you would collect them using trypsin, acutase, or EDTA, which will result in remaining aggregates. Very few, I know just a very few groups that really digest to single cells. Most of them keep aggregates, also for treatment purposes, by mistake or by purpose. So these are, the first one here is trypsin treated, and that was done at Life and Brain in Bonn for us. Because we wanted to know how big single cells are of iPSCs, and you see they're roughly 14 micrometers in diameter. And based on this information we got working with them, we have now one standard protocol to measure iPSCs. And the good news is for those that work with the cells, you don't have to care if you digest long, you digest short, you don't digest, use EDTA for collection or whatever you use, and whatever remaining levels of aggregation you have, the CASY will analyze for it. So you can even work with high levels of aggregates. Don't worry, you have the result. So that makes handling easier. And for those that are interested, and if you are interested, for those from working with stem cells or other things, I may have some slides even later on another system, which is an incubator. But we can decide if you want to see or not and how much time we have left, where we are using defined cell numbers to start with, and especially on iPSCs, CASY is the only one that gives reproducible results, so we can standardize, and which is especially important for big pharma customers we have, but also for smaller ones like academic and so on. This is an overview, generally, what we can do. So we have 3 different capillaries. Once they're calibrated, you can exchange them yourself. So no service technician is needed for that. And we have also the size range here. So for bigger cells, medium size, smaller cells, and what we recommend to use it for. So it's quite reliable. And for example, this is a guy from German Cancer Research Center, which is probably the biggest institute in Europe, where they measure, I think, 50 or 100 different cell types a day. So it's quite heavily used. Now, the question was about PBMCs. For those that are not aware of PBMCs, what you do is you have some density solution, let's call it that way, and you overlay it with whole blood from a donor. Then you centrifuge for half an hour. And then after that, you have basically the red blood cells and the majority of the granulocytes below that, let's call it cushion. And then you have the lymphocytes on the other side, and then the plasma on top. So this is the fraction, the yellow one, that you're interested in, and it contains the peripheral blood mononuclear cells. The problem is that when you count, you will get a total count number, more or less precise. But what you will not have is the ratio between the different cell types. And to come back to an earlier question, here you see there are in CASY 2 peaks, and we can analyze up to 5 different dependent areas. So we can have an area for the dead cells, we can have an area for the lymphocytes, for the monocytes, and maybe other cells. And what you see here directly, because this is a problem that you have very often, the ratio between lymphocytes and monocytes may vary dramatically, depending on the donor. And with CASY, you can already have this information just with a simple measurement. So you have a total cell count, you have viability, but you also have the concentration of lymphocytes or monocytes. So that is also a possibility. This is published data. This group is using heavily the CASY as well for analyzing T-cell activation. Why is this interesting? They love the instrument because it can track the proliferation and the size growth simultaneously. Both are required for activation. And here you see the normal status. So you have inactivated T cells, and then after 24 hours, they already grow significantly. And another 24 hours later, you see that the cells are getting heterogeneous and have grown even further. And in this paper, they were simulating microvirus environment. So, and to see like absence of glucose or presence of lactic acid. And they were analyzing what the impact of those situations is on activation of T cells. Said it's available as a paper. Now we come to insect cells. This is baculovirus system. It's another group in Germany. It's a biotech company. They are using this for 20 years now, and they are interested in, so they infect SF9 insect cells with baculovirus, and then they want to stop the process before the cells burst. And this is a standardized process which is completely dependent on the CASY, and they're using it for, as I said, for 20 years now. There is another one. Now we come to yeast. In this case, it's brewery yeast. And it's a very nice example that CASY can warn you in case of contaminations. You saw the schematic one, which is easy if you have big cells and you may have a separate peak. But even if you look at yeast and you have a yeast infection, you will get an indication. And this is normal lager yeast, so Saccharomyces cerevisiae, quite robust. You see the green line and the green size distribution. And what they do is they ferment, then they have the so-called harvest yeast at the end of the process, and they reuse it. And one time I got this hold of this one where the peak looks different. You see clearly there's something going on. And when they analyzed it in microbiology, they clearly could see that there was a wild yeast contamination. So just by looking at the size distribution can give you information of contaminations or other changes, which is quite important. Not sure if anybody works with algae, but even there you can analyze with CASY the cell cycle. So this is a sweetwater algae, which is half donut shaped. In this experience, it was synchronized by light, so it starts budding at the same time. And what you can see is at the beginning, they're all 6 micrometer big. And then when the budding starts, you have the single buds, And then the 4 buds disappear. Then you have 3 buds left, 2 buds, which we could not show because this single peak got too high. And then the whole cells grow in volume again. There, between the cell cycle detection, we also had an EC50 detection, which is commonly used in environmental testing. I heard that a lot of people are working in bacteria. What to say about bacteria? 2 things are important. It's fast, so 45 seconds normally. You only have a cup, you pipette your sample in. Sometimes you have to do pre-dilutions, but that's it. And other than that, it's probably the most simple sample for CASY. Normally, we would count about up to 50,000 cells per measurement, so also a very representative sample compared to like plating with 100, 200, 300, depending what the number is. It works with very different kind of bacteria. So if you're interested in, there are also a lot of publications using CASY. The most complicated and ugly one that I am aware of, at least from those I've seen, is Leptospira. And also because of the shape, but also there are some which are heavily aggregating. So it works with all of them. And I just have some very common one like E. coli or Bacillus subtilis. So it works with different shapes, whether they're coccus, whether they are rod shaped, whether they're spiral. Yeah, nothing to worry about. And just if you use a CASY, 45 seconds later, you will have the result. One thing we need to mention is, since bacteria don't have a nucleus, we can only detect viable cells. So as soon as the cell membrane is gone or defect, the cells disappear. So we detect the viable cells. However, we also can detect VBNCs, viable but not culturable. So cells which are viable but which do not grow. Here, this is a very loyal customer from Japan. Most of the work was done in Zurich, but now he's back in Japan, just on pollen. It's more a niche, but also because we're so independent of shapes, of forms, and it's quite fast and reproducible. And then finally, there's some other ideas. What we do is, in case of fungi spores, this is an example where we differentiate between haploidic and diploidic spores. Then we have, it seems to be very popular these days, is blood parasites, trypanosomes, leishmania. So we have a lot of customers currently buying the instrument for this application. I think there was a question earlier about blood. So the orange one was detected with a 3 micrometer capillary. The orange one is full blood. Obviously, there is a high amount of red blood cells detected. And then after centrifugation, the blue one, which contains the inactive and active platelets. Finally, these are particles. So these are NIST certified particles for size. This was the certified size. This is what we measure, and this is actually the size deviation. I don't want to run through this, but I think what you hopefully could see is CASY is probably the most accurate thing you can use for counting. You can simply rely on it. We have many customers, like we just did an analysis of CASY publications from 2017 to today. We usually find, and we know we don't find all papers, but we find those where a lot of them, we have about 100 to 130 publications a year, which we find. It's all over, from bacteria to PBMCs to cancer to stem cell research and so on. Interestingly, the impact factor of all those publications on average is 6 and above, so it's quite good ranking. We have very loyal users. We just had some people that went from Germany to US to Harvard Medical School or MIT, and they also bought a CASY there. And these people did just recently 3 full Nature papers. Yesterday, we visited a guy from DKFZ, German Cancer Research Center, which has just 2 or 3 Nature papers this year, plus some others, Nature.com and so on. So it's really like, if you're serious about what you do, CASY is the one you can rely on. So it will not let you down, and will give you perfect starting conditions so that you have completely your experiments under control from beginning to the end. Good. Questions?
Thank you, Dr. Joachim, for this amazing sharing. I'm pretty sure that there will be some questions from our participants. Can we start the Q&A session now?
Sure. I mean, for those that are interested, I think we were planning 1.5 hours. I thought that there are some people looking at iPSCs, at MSCs, and some other things. If some volunteers are there and they can write it maybe in the chat, I can quickly show to you a very novel incubator, especially for those, where we have just this year extremely good result from pharma, which means to differentiate to organoids, spheroids, but even also prevention of tissue. But just that you know that, or we make a separate session if you're interested. It's a game-changing instrument, to be honest, and I'm not so often saying things like this. But first question on CASY. Very silent.
Yeah, or is there any questions?
Hi, Dr. Joachim, Dr. Pavel. I'm sorry. I'm Alicia.
Joachim is fine.
Yeah. Hi. May I ask, how is this different from flow cytometry?
The detection method. So we are, I mean, in principle, we only measure current. And while the flow is using is looking at light diffusion of light, basically. But we both measure in 3D in a flow cell. So that's the advantage that both instruments share. The CASY doesn't. The first thing is sample preparation. So the CASY will not require you to do anything special. No antibodies, no staining. So you just take the cells or the sample as you have them, and you give an aliquot into a cup with CASY toll. Usually 10, 20, 50 microliters or something like this. Then you mix it quickly and you put it to the instrument and measure. So it's a very straightforward process. It goes fast and doesn't need anything sophisticated. The only thing you really need to do is precise pipetting. So if you pipetting is a problem, then yeah, okay, then that's a weakness. And then the measurement on a flow is typically around 10 to 15 euros. Here, it's in the area of cents. So very different there. And it's also, we have customers where 50 people use, in big institutions, where 50 people use the CASYs, and it's very easy to operate. And on the other side, it's very difficult to make mistakes with it.
Okay, thank you. Thank you.
You're welcome. Other questions?
Any questions for Dr. Please do ask if you have any questions for Dr. Joachim.
Okay. Anyway, you can follow. I mean, you can talk with my partners in Malaysia anytime, and I think we will be happy to answer all your questions. And also, maybe if you share what you're doing, we can get even more dedicated information for you.
Sure, for sure, we will help. Thank you, Dr. Joachim.
Thank you very much.
It's really a comprehensive presentation for you. Thank you for joining.
Thank you very much. And please feel free to contact us and with any questions you have later on.
Sure, Dr. Joachim.
Okay, bye.
Thank you. See you again. Have a nice day.